Can I just move around? And,
Okay.
Like,
All right, so in this slide, this is respiratory epithelia. We're going to see a transition from the trachea here and the pseudostratified ciliated columnar cells that are seen at low magnification right here that line the larger airways like bronchi and the actual trachea itself here into these smaller branches, the primary, secondary branches that we find here. That will transition all the way as you make your way through the respiratory pathway. It'll transition into the deep areas of the lung and eventually become alveoli with simple squamous line. That would require the understanding that as we move into these squamous cells and the type 1 alveolar cells of the air sacs, we're going to see a transition throughout the bronchial tree of pseudostratified ciliated columnar epithelia to simple columnar epithelia in the smaller branches here. And then that'll lead into simple cuboidal lining, which will then move us into stratified squamous and eventually into the simple squamous of each alveolus. The slide that we're looking at here is primarily used to show the layer of epithelia that allows for diffusion of respiratory gases between a capillary, a small capillary like this, and the air sac that it is adjacent to. And that pair of structures was referred to as the respiratory membrane. Okay, this is the kidney slide. What we're looking at in this picture here and in this picture right here is a sort of a larger view, 60x view of one of the medullary region, the medullary pyramid in the kidney. This outer region here is the cortical region where the majority of the glomeruli are found, and this is a low magnification view of each of those glomeruli in that cortical region out here on the outer edge with a number of different kidney tubules, cuboidal line kidney tubules surrounding it. This would be deeper into the medullary region right here. We can see a nice blood vessel, and that blood vessel would contain a number of blood cells surrounding it, areolar connective tissue, and that would be binding the tissue we've previously described with the glomeruli and the cuboidal line tubules surrounding it. This vessel would be located in the kidney as between those two portions. This is a good picture that we're looking at of the glomerulus. So this is one of these magnified at full magnification, and we would be looking at the simple squamous cells in a chain all the way around the Bowman's capsule, and that capsule is seen here with its squamous cells as the structure that would receive filtrate as it's filtered through the capillary bed or the glomerulus itself and the endothelial lining that's seen here under hydrostatic pressure. This is that glomerulus that we were just pointing to. This is the cup that receives the filtrate, and then diffusion would occur across the simple squamous membrane into the interstitial or extracellular matrix that's in between kidney tubules like we would see right here. This is that structure also. Here's the tubule. This is the capsule or cup that receives the filtrate after hydrostatic pressure pushes it from through the walls of the glomerulus or capillary bed into Bowman's capsule. Okay, this is also a kidney slide. The same structure shown is just a different view at a different level of the kidney that shows obliquely kidney tubules that normally are quite circular in nature as they move through that area. Here are some of them at lower magnification that are circular, and if you cut it at an oblique angle, many of them might appear as an oblique structure like we see here, or fully magnified right here. Here, of kidney tubules are lined with a simple cuboidal epithelial tissue that has the ability to secrete or absorb the filtrates materials in and out. Okay, this is in the small intestine and digestive tract. This is probably right here is the best microscopic depiction of these columnar cells in this location. A basement membrane down here closest to the nuclei would be present. We would have a number of different structures below this lining. For example, this would be a cuboidal lined submucosal duct of a gland. and then we would have a number of different types of cells throughout the areolar tissue that it's bound to. You can see different numbers of goblet cells associated on those apical surfaces, producing mucus to decrease surface tension as you move through the digestive tract and the tubing that's carrying the waste. And as this occurs, you're going to see a decrease in the moisture level of that feces that's being produced. And as you move it through, especially the large intestine, goblet cell number would have to increase. The brush border would have to disappear, and just water being removed from the lining, or from the tissue that's moving through the lining, or the waste moving through the tissue's lining, sorry, you would have the need to increase mucus production to lubricate that tube and decrease surface tension so that the waste can arrive in the storage area of the rectum. Within the folds that are seen here throughout the digestive tract, each of these folds are the actual villi. Each of the villi are seen up here at a low magnification, and on the villus itself, you're going to find, as you magnify that, you would find a dark line or a brush border seen here on that apical surface for increasing the absorptive surface area and the absorption of nutrients that are required from the food that's been eaten. As that food moves in, or as those nutrients move in through that brush border and into these villi, you're going to find right here in the center of each villus, okay, there's what's referred to as a lacteal, a lymphatic structure that absorbs those nutrients and sends them directly to the bloodstream if that passageway isn't a direct line or link. That's also seen here in one that's laid down sideways. This is a lacteal as well. Okay, so these cells that we're looking at, to reiterate, are columnar. Their primary function is absorption, and the line of microvilli, or the brush border on their apical surface, enhances, thousands of times the ability of these cells to absorb the nutrients from the food we've eaten. This is a view also of the bronchial tree that we were looking at before. It's just a closer look at the lining of the airways that lead into the simple squamous lined air sacs or alveoli. So as we showed before, as the air moves in to our airway or our voice box, the larynx, we're going to bring that air through the trachea. And the lining of this area that leads into these branched portions that eventually lead into the alveoli consists of pseudostratified ciliated columnar epithelia. This type of epithelia would be pseudostratified because the false appearance of stratification, or the double layer of nuclei, is the result of the placement of the numerous goblet cells, the unicellular glands producing mucus, so that the air that we're breathing can be cleansed and rid of debris and any dust particles that would need to be eliminated. The cilia that's present here all throughout is visible here. Okay, the cells at the basal membrane, or the basement membrane, the basal lamina, since this is a mucous membrane, would be more cuboid-like because they're mitotically dividing, they're younger, and they haven't quite reached the size and the quantity of the colloid cytoplasmic material within them as a mature cell would have, seen here. As we look at this tissue at a low magnification, it's important to note that this is an excellent way to view multiple types of tissues, just about all the tissues at once, is seen from the inner lining, where you would have, of course, the epithelial tissue, this tissue, the pseudostratified ciliated columnar epithelia, and the numerous different goblet cells that result in the pseudostratification or pseudostratified appearance that we've talked about already. Just below the basement membrane, or part of the basement membrane, or lamina propria, we would see where the cells are actually sitting on top of that structure, which is the basal lamina here, seen in this drawing. And then beneath that we have areolar connective tissue, also seen here, and that band of tissue is seen easily here as the double part structure that's going to be identified as the lamina propria. This is mostly identified primarily in the mucous membranes that line our body's moist surfaces or entryways. Down below the area where we have the submucosa, so that's the mucosal lining. Here's the submucosal area that has a great amount of arteries, veins, and lymphatic ducts of glands. We have the layer of hyaline cartilage that forms the C-shaped rings seen here in the airway that is our trachea. Those cartilaginous pieces would continue all the way into the tertiary bronchi, or the third level of branching here, and those would become bronchioles, would lose the cartilage and would increase smooth muscle to reinforce that airway to the point of alveoli in the transition. This is our perichondrium. Since cartilage, hyaline cartilage, is an avascular tissue, and the chondrocytes residing in the lacunae throughout are going to need a supply of nutrients and blood gases and elimination of CO2. We would surround this hyaline cartilage with a band of dense irregular connective tissue to provide what it needs, and that's referred to here as the perichondrium of that cartilage. Then as we move towards the outer surface, if this is the inner lining, this is our airway, this is the middle of this tube. As we move out towards the surface of the neck, or out this way, we would hit next the hypodermal layer, or the subcutaneous layer of fat, and then from there we would move to the thickest layer of the skin, the dermis, and then further out to the outermost layer, the epidermal layer of the skin, the keratinized stratified squamous epithelium. This is a great picture showing each of the mucus-producing goblet cells throughout the apparent double layering of nuclei produced for the reasons we've discussed already. Here's a nice view in the submucosa of a simple cuboidal-lined duct of a small lymphatic gland. You can see on the edges of the cartilage, further magnified here, when you look at this, you see chondroblasts that are differentiating, pushing as they mature, leaving that differentiated new chondroblast in its place, and then maturing and moving to the center in its lacunae in a rigid matrix that contains GAGs, chondroitin sulfate, hyaluronic acid, and water as the matrix that's hardened and resilient to any stress or strain that's placed on it. In this picture we can see both keratinizing and non-keratinizing stratified squamous epithelial tissue that's going to either line moist body surfaces, most moist body openings like this tissue would, versus the layering of dead cells, the keratinized stratified squamous, that would sit on the surface of our skin that covers our entire body in the integumentary system. If you see dead layers of cells on the outer apical surface here, regardless of the thickness, then you're looking at a keratinizing layer, or the stratum corneum on the outer surface of the skin that protects our body from water level changes, or keeps us waterproof. It also protects us from abrasion and from any damage that might occur externally. These layers again are dead and are constantly sloughing off, and are referred to as the keratinized stratified squamous epithelial cells that result from the keratinocytes dying off at this layer. We'll mention that in a moment. This layering right here and this show all living cells. So here is our basement membrane. This is a moist body opening, so this lamina propria structure is going to, as we can see down here below, consist like it did in the trachea of basal lamina and a reticular lamina, a non-living double layer that makes up the lamina propria in moist body openings or mucous membranes like we've discussed. These cells are also all non-keratinized stratified squamous epithelial cells. They are all living, and they're what we obtained cheek cells from that we stained in Lab 2, and we stained with methylene blue and viewed in the microscope for the first time in this class. This picture here shows a great view of the different strata that's seen throughout the stratified epithelial layers. Notice, as we've stated before in previous slides, we find the basal cells are more cuboidal, And as these cells divide and push out older cells or replace themselves and then move into this layering that we see here, they will become flattened and eventually, if it is in a skin layering, and these cells will die off, be crushed, release keratohyalin granules, combined with lamellated granules that provide a means of prevention of water loss from our surface. These two cell layers are, again, thick skin. This is in moist body openings. The next slide that we look at will show these two separately. Every picture that we're looking at in this slide is showing the non-keratinized stratified squamous epithelial layers. Every layer of cells is living. This shows a nice depiction of those mitotically active cells, and this as those cells are pushed out and mature, they're flattened, but every layer is living. Each of these examples are different magnifications of our moist body openings, linings, for protection against abrasion in those areas. In this picture, we can see that we're looking at three examples of different thicknesses of keratinized stratified squamous epithelia and a fully magnified view of that same tissue, so 4 examples. This is a picture of thin skin with 4 basic layers. This is the stratum corneum. The last living layer of cells here would be the stratum granulosum. This outer layer of the corneum, by the way, is approximately 20 to 30 layers of cells, dense, or dead cell layers, thick. This would be 3 to 5 layers, the stratum granulosum. Stratum spinosum beneath that would be the thickest layer of living cells in the skin, and would be approximately 2 to 12 to 15 layers of cells thick. These cells, by the way, are keratinocytes, and we would find at the basal layer, the single layer of cells at the basement membrane here, we would see a combination of 2 different types of cells. One cell type that's producing the pigment melanin called melanocytes, and the other abundant type of cell we find here would be the keratinocytes that have been previously mentioned. Keratinocytes are the only cells that can leave this bottom layer and be found in each of these layers outside of the basal layer. This is thin skin and would often be associated with hair follicles, since we are mammals. And this is also thin skin. Here's a great view showing the waterproofing that occurs throughout the body surface in this type of tissue. As we add or we look at these 2 types here, we've added a 5th layer to this tissue, and you can see that there is no hair follicles present. There is a 5th band or layer of cells right here called the stratum lucidum, and therefore we have stratum corneum, we have stratum lucidum, both dead layers of cells, then the stratum granulosum, stratum spinosum, and the stratum basale or germinativum, all the way deep into the epidermis, that will be sitting and coupled with the dermal papillae from below and joining from the epidermis, the epidermal pegs with that dermal papillus. Each of these pictures are excellent views of keratinized stratified squamous epithelial tissue. These are rare examples of tissues that we're looking at here. These are stratified cuboidal or stratified columnar tubules, or in this case the duct of a large salivary gland. It's a low magnification, but you can definitely see the multiple layering of cells that's occurring that are cuboidal here as well. The same type of presentation of cells, and then one of these structures cut in cross-section that's leading into this larger salivary gland, further magnified, would appear as this stratified epithelial type that we see here. A fairly rare occurrence though, and it's mainly for protection in from the secreted substance in the gland that it is associated with or leading away from.
after we do this next stratified one, we could do the other ones tomorrow.
Okay, we're rolling though.
Yeah. I mean, we're good. We're good. I just want to take it.
Cool. Okay. Here's an even rarer type of tissue, the stratified columnar type. You can see some goblet cells. This too would be found in the salivary ducts, protecting against some of the components of saliva that's being produced and secreted into this tubing. So also a very rare tissue, very rare occurrence to see cuboidal or columnar stratification. And this is, since it's so rare, wouldn't be a primary focus. This is sort of what we're trying to teach is the norm, and this is a rarely found tissue. It's only in certain parts of certain areas of the body. The last stratified type of tissue we see is within the urinary structures, the urinary bladder, the urethra, and the ureter that leads into that bladder. You can see that when that bladder is empty, you're going to have a folding that occurs in the inner lining, and as the bladder is filled through the ureter and the filtrate accumulates, you're going to see a transition from these cuboidal cells in an empty bladder to a stretching or a distended wall, and that transition would be to a more squamous type appearance. You can see a nice view of the basement membrane here that's occurring, and then approximately 4 or 5 layers of anywhere from cuboidal to columnar type cells that then become squamous in a distended bladder. Like we see here, it's just difficult to show a full bladder on a slide if you want to keep things dry. All right, so that's transitional. One more time, it's a transitions from a cuboidal or columnar appearance to a flattened plaque building appearance of flattened cells that looks like this, and a protective, distended or stretched wall of the bladder when it's full. You want to keep going? Okay. Underneath those epithelial linings, here we have what we were just looking at. This is a great view right here of the skin that covers the surface of the body. And you can see keratinization here and the dead cells sloughing off. This looks like and I'm not sure where it was taken from, but it definitely looks like, because of the layers here, it's been cut from superficial to deep through the layers of the keratinized stratified epithelia. Keratinized being dead layers, so the surface of the body. We'd move through the dermis, the thickest layer of the skin, into the hypodermis here. We see a number of large blood vessels. There's an artery, there's a large vein, and the adipose connective tissue that makes that structure, the hypodermis or subcutaneous layer up, and skeletal muscle just beneath that as we move in towards the skeletal muscle's attachment to bone. This is a great picture showing the same sequence of cells and tissues as we move inward, and the bundles of muscle that would be here present in that slide. Now, what we focused on in this slide is the areolar connective tissue, or the two-sided tape that's forming the binding and support of adjacent tissues that are unlike one another. So underneath the epithelial tissue here, we would find this tissue, areolar, in between any structure that's different from the denser regular of this layer, like these two blood vessels, the artery here and the vein, and if that has around it a different kind of tissue, which it would, this artery would be surrounded by a muscular, smooth muscle wall, as would this large vein, but it wouldn't always have muscle if it's a vein. But this vein would have the need to connect with the tissue around it, and the binding support that's necessary here would be provided by the thin layer of two-sided tape, or areolar, that links this vessel to the actual fat that it's depositing as the blood is delivered here. There's a number of different cells in each of these slides. The most numerous type of cell would be, of course, the fibroblasts, the smaller conical-shaped cells here, and fibrocytes that are the mature version of that, laying down the matrix of hyaluronic acid and water throughout the ground substance that would be added to hold together the fiber types, the collagen fiber, the elastic fiber types, and then the reticular fibers that are present in this particular slide. It also provides a means for transport and movement throughout two separate types of tissue of vascularity and nutrients or blood gases coming and going between an avascular epithelial lining that we just talked about here and the highly vascular dermal layer beneath it. These are both larger cells, mast cells, releasing histamine and heparin, involved in the inflammatory response and in thinning of the blood during that increased flow of blood throughout the body, which would be the first response for any tissue repair or damage in the body. This circular cell is a white blood cell or leukocyte, and we'd also potentially find in this particular slide larger macrophages and fat cells as well as plasma cells from the blood that's circulating through it. This is adipose connective tissue, and it'll be continued soon. Cool? And then I don't know what time.